Hi, today I will be providing a brief overview of protein-ligand docking, a form of molecular docking used commonly in computer-aided drug design. Docking helps reduce the time and cost of drug discovery by computationally prioritizing compounds most likely to bind to the target before experimental screening. The shown graphic represents rendering by Schrödinger of work by the Lee lab on sabuzabulin, an investigational microtubule disruptor. Shown in green is the co-crystal structure of TN16 bound to the colchicine site of tubulin, which was used to retrospectively rationalize the binding mode and mechanism of action of sabuzabulin via docking after its discovery. Protein-ligand docking seeks to fundamentally answer two questions. Docking methods predict and score binding poses of ligands, which is the orientation and their conformation of ligands within protein binding sites, which provides insight into interaction energetics and relative binding favorability. These results can answer a variety of questions about virtual screening libraries, lead optimizations, as well as related topics. Importantly, docking does not simulate binding kinetics or pathways and instead finds energy local minima. The development of protein-ligand docking stems from, in parallel largely, the hypotheses on the liganding of enzymes. The lock-and-key hypothesis in 1894 was derived from Emil Fischer's discovery that glycolytic enzymes could distinguish between stereoisomeric sugars. This theory proposes that enzyme specificity arises from geometrically complementary shapes. This model assumes that both the enzyme active site and ligand are rigid before and upon binding. Such is analogous to early docking methods that assumed a rigid receptor and rigid ligand for low computational cost. Koechlin proposed the induced fit model in 1958, motivated by two observations. First, 5-nucleotidase does not hydrolyze ribose 5-phosphate despite the substrate's small size fitting the geometric requirements of the active site for 5-AMP. And second, that a rigid enzyme model of templated protein synthesis would require an unconceivably large number of specific enzymes for amino acid condensation. In the induced fit model, substrate binding to a flexible active site induces a conformational change that correctly positions both the substrate and catalytic residues for catalysis. The most recent model, formalized by Nuzanov and colleagues in 1999, based on work on protein folding and conformational energy landscape, proposes that ligands selectively bind pre-existing sparsely populated enzyme conformations and shift the equilibrium toward the bound state, even if that conformation is not the most stable. We now know that true enzyme ligating is a combination of the following models on a continuum. The docking workflow requires preparation of both the ligand and protein prior to docking. Smiles are gathered and all reasonable charge and tautomeric states are calculated, and each is carried forward into a 3D structure generation and conformational energy minimization. Protein preparation begins with the crystal structure from the PDB, which often contains crystallographic artifacts such as buffer salts and may lack resolved coordinates for flexible loops or disordered regions. These must be modeled. Hydrogens, which are invisible in X-ray crystallography, are added. Residues are protonated at the desired pH, and constrained energy minimization yields a docking-ready structure. Depending on the docking approach, the active site may then need to be explicitly defined. With both prepared ligands and protein in hand, the docking engine generates a series of sampled poses with associated scores. Poses are then filtered and clustered before evaluation based on score and chemical reasonableness of the predicted binding interactions. A ligand has 6 degrees of rotational and translational freedom, plus additional degrees of freedom with each rotatable bond. Sampling all possible poses would be computationally unfeasible. Pose sampling methods, therefore, search through possible positions, orientations, and conformations a ligand can adopt within a binding site, with varied degrees of flexibility applied to the protein and/or ligand. Rigid ligand docking was the earliest approach due to its computational simplicity, but is largely historical. Semi-flexible docking by a flexible ligand, but not protein, is now dominant as the balance between accuracy and computational cost. Fully flexible docking is the most physically realistic, but does not necessarily improve accuracy, as the expanded conformational space increases false positives and exposes scoring function limitations. Docking is most reliable when the target structure comes from co-crystallization with the bound ligand, as empty pockets can collapse and distort binding geometry. Accuracy further improves when the co-crystal ligand is structurally similar to the compound being docked, as the binding site is then pre-organized in a conformation complementary to that chemotype. There are a variety of different softwares available for ligand-protein docking, and I have listed just a few here. Sampling algorithms and scoring functions represent two large sources of variations across softwares. The slide lists several sampling approaches. Stochastic methods, which rely on random structural manipulations, such as Monte Carlo, and genetic algorithms, tend to be widely used in modern softwares. For the sake of time, viewers are directed to the review at the bottom for a more comprehensive discussion on the different approaches. There are 4 predominant classes of scoring functions used in docking. Force field scoring is the most physically grounded, using classical mechanics to model non-bonded interactions such as electrostatics, van der Waals, and hydrogen bonding between paired protein and ligand atoms. Due to computational cost, however, long-range interactions are truncated by a distance cutoff, and solvation and entropic contributions are poorly treated because these methods do not account for explicit solvent-solute interactions and water reorganization upon ligand binding. Empirical scoring is computationally cheaper and uses chemically intuitive interaction terms such as H-bonding, hydrophobic contacts, ionic interactions, as well as rotatable bond penalties. Parameters are regression fit against experimental binding affinities of known protein-ligand complexes. However, these methods extrapolate poorly outside the chemical space of training sets, struggle to account for cooperative terms, and provide values highly variable on software. Representing another scoring function family, knowledge-based scoring is derived from inverse statistical mechanics and uses the frequency of interatomic contacts from crystal structures as an indicator of energetic favorability. Such suffers biases from proteins that are much more readily crystallized, and interactions poorly represented in the PDB, often including metals, halogens, and unusual heterocycles, are poorly calibrated for. Lastly, there's a growing effort to include machine learning as a means of scoring. Machine learning scoring functions train neural networks directly on structural and affinity data, and learning interaction scoring without predefined physical or chemical terms. Like the others, this suffers from inaccuracy when extrapolated to chemical models outside the training set. To highlight the power of docking, I would like to talk about this collaborative effort between the Irwin, Shoiche, and Roth labs published in 2019, in which they worked on using docking of large libraries of products that could be synthesized on demand from enamine's building block library, as shown in this large figure here. Towards the synthesis of AMP-C beta-lactamase inhibitors, they applied a workflow that involved docking 99 million of these molecules and testing 44 of 51 selected hits that represented distinct and novel compound clusters. Ultimately, they identified 5 active hits, in which one was such scaffold was further optimized to a 77 nanomolar scaffold, which possesses a phenol warhead, a novel chemotype in this antibiotic space. Interestingly, the docking poses were able to identify not only the desired binding pocket, but also with low RMSD compared to co-crystal structures as shown below for several phenolic triazole scaffolds, with purple representing the various compounds and blue representing docking results. Such screening of this compound library scale would be otherwise too labor-intensive to test experimentally. In conclusion, docking is a powerful computational tool for predicting ligand binding poses within a protein active site, using sampling algorithms and scoring functions. While the scoring function limitations and protein flexibility remain key challenges, docking has proven invaluable for prioritizing compounds in drug discovery. Thank you for listening to my talk.